Oh, right, lecture. Sorry about that. So here we are, lecture 4. So for this week, we're past the halfway point after today. Hope that you've been taking care of yourself. I've been just enjoying some snacks, and I hope that you have as well. So we're continuing on with our neurophysiology lecture series, and we're going to jump right into it because we have a lot of ground to cover over the next 29 slides. I hope that everything has been going well for you so far. I know that you have a lot on your plates this week, but I hope that you'll be finding these lectures going at the pace that you need. And if you need me to slow down or you need me to speed up, that you'll let me know about that. So I'm going to go ahead now and start sharing my screen with you all. So just give me 1 second as I get that up there for you. And as I switch over to the laser pointer. And I'm hoping that everything is going to work well for all of us today. So we're going to go back and talk a little bit in episode number 4 about what we call the spike conundrum. So we've been talking about action potentials, and we've been talking about how they work, the different things that make action potentials more effective, the things like the axonal diameter, the things like myelination, all helping to speed up the conduction of these action potentials. And we last left off looking at what happened to the action potential when it reached those terminal boutons, and it caused the release of neurotransmitters through either that full fusion event or through the kiss and run event. And now what we're going to do next is to look at what happens after the neurotransmitters leave. And here we're looking at what happens when the signal gets sent to the next cell. And we're looking at these postsynaptic receptors today. And we're going to identify the different classes of these postsynaptic receptors. We're going to identify them as being either ionotropic or metabotropic. And then when they get activated, because we're not activating voltage-gated sodium channels, we're going to see how this postsynaptic potential that gets generated, how it's actually going to spread across the surface of that neuron, and hopefully through different mechanisms like PSP or post-synaptic potentiation, if we can sum a lot of these signals together from many different neurons, like spatial summation, or we get one neuron firing consecutively, temporal summation, that maybe we will be able to cause this next neuron along the way to also fire an action potential. And that summary is given to you over here. So for example, if we stuck in an electrode into this postsynaptic cell, the one that we're interested in now, up until this point in time, we've been examining all of the events that occur presynaptically. Can we reproduce that? And can we get this next neuron to do exactly the same thing? So here it is, sitting at rest, minus 70 millivolts. This is sort of the resting membrane potential. We've now seen the release of these neurotransmitters through the voltage-gated calcium channels and the activation of that kiss and run or the full fusion event. The neurotransmitters get released. And as the neurotransmitters now leave and they bind to these postsynaptic receptors, can we actually measure these different changes that are occurring in this postsynaptic cell? And that basically is what we're going to be talking about today. I'm hoping that it's a clear roadmap for you, and that looking at these postsynaptic receptors will be something that will make more sense in terms of the context of the previous lectures. So when we last left off, we were looking at these vesicles, and now this transmitter agent or these neurotransmitters are going to be released, diffuse across the synapse, and bind to these postsynaptic receptors that I just showed you on that previous slide. And these are embedded in that postsynaptic membrane, the next neuron over, and the binding of this neurotransmitter that has just gotten released is going to cause a change in the shape of the receptor protein that it's going to interact with. And these receptors are generally fall into two of these different classes. They're either ionotropic, where the neurotransmitter binds, and as it binds, it directly opens up a channel, or we're going to see that they are metabotropic, where after the neurotransmitter binds, it will initiate a metabolomic change or cascade to activate various enzymes. And we'll see that it's really the receptor that determines the effect that we get on the postsynaptic cell. It's not necessarily the transmitter. Confusing statement, right? So bear with me for the time being, and come back and circle back to this last bullet point to make sure that it's clear for you. All right, so let's just dive right in. Let's look at one of the first classes. This is the one that I studied as a graduate student over at the Hospital for Sick Children. I was interested in ionotropic glutamate receptors. And here we know a lot about these different types of receptors. We know that when a ligand, often the neurotransmitter, so a ligand or neurotransmitter binds to this channel, we call this an ionotropic receptor. So the binding of that transmitter to the postsynaptic membrane is going to bind to this receptor that's found on that membrane, and it's going to result, after binding, in a postsynaptic membrane potential, and we call this a PSP. And depending on whether it's excitatory or inhibitory, we can call it an EPSP or an IPSP if it's inhibitory, and this is that PSP that we're looking at. The duration of these PSPs, on average, a single event is about 20 to 40 milliseconds in duration. And it's usually short because the neurotransmitters aren't staying there continually. We're going to get to that in lecture number 6, so bear with me. And as long as the neurotransmitters are there, and they are there for a short duration, these channels are going to be open. The ion channel that is associated with that receptor is going to be specific for a cation. So for example, if we had an ionotropic receptor that bound to things like glutamate, it's not going to let in any ion. It will then open up a channel that would be specific for things like sodium or for potassium. And because the movement of sodium inward and potassium outward would cause net depolarization, we would say that that particular ionotropic receptor generated an EPSP, or an excitatory postsynaptic potential. If, on the other hand, a neurotransmitter bound to its receptor, like a GABA receptor, and it would then open up that channel, often that channel that is found in that receptor, which is just a pore, it's going to be selective for different anions like chloride, or it may lead to the removal of potassium. It will flow out of the cell. In either case, it will cause inhibition, or it will lower the membrane potential and make it more negative, and we call this an IPSP, or a hyperpolarizing postsynaptic potential. Okay, so I'm hoping that's clear for you. I'm going to use those terms quite frequently, so understanding the difference between EPSP, IPSP, and these ionotropic effects is going to be really important. So the classic type of ionotropic receptor is illustrated for you on this particular diagram. And this is actually the first characterized ionotropic receptor. It's the granddaddy of all of these ionotropic receptors. And this is called the nicotinic receptor for acetylcholine. And it's often abbreviated as small n. Acetylcholine is abbreviated as ACH, and we use R for receptor. So a nicotinic receptor, or a nicotinic receptor for acetylcholine, what will happen is acetylcholine, a neurotransmitter, will be released. It will then bind to these postsynaptic channels, which are normally closed. And when the receptor binds to the neurotransmitter, it will cause a conformational change. And this will then allow for the influx of sodium. It will also cause the efflux of potassium. And as a net result, this cell will become more excited or more depolarized. Hope that's clear. And again, this is a classic example of an ionotropic receptor. There are a number of different ligands, or things that will bind to these ionotropic receptors. And these could be different types of neurotransmitters. And the ones that we see most often, and these will directly cause the movement of ions, we will see receptors for acetylcholine. So the nicotinic acetylcholine receptor is a classic example. And it's called nicotine, by the way, because the drug or the substance known as nicotine will bind to these same receptors. We don't have nicotine in our bodies, but when we, if someone were to inhale them, it would be acting on these nicotinic acetylcholine receptors. And it's just a pharmacological way to identify it. In addition, we have glutamate ionotropic receptors. We would have GABA receptors, and again, GABA being a neurotransmitter. Glycine is another neurotransmitter that will bind on and cause ionotropic receptors to open. Just keep in mind, though, that these same substances, acetylcholine, glutamate, GABA, as well as glycine in some instances, can also cause metabotropic receptors and bind to them and cause metabotropic receptors to become activated as well, and they may have completely different effects. So this is what it means when we say that it's the receptor that determines what happens to a cell, not necessarily the neurotransmitter. Just because glutamate got released, it doesn't mean it's always going to cause excitation. If it binds to an ionotropic receptor, it will cause excitation. But if it binds to a metabotropic receptor, it may or may not cause excitation. So I'm hoping that that earlier bullet point is now a little bit more clear for you. All right, keep jotting down those notes. Keep track of things that are confusing for you or things that are not so clear. I'm going to try my best to answer your questions as we go along. All right, so then there's this other class of receptors that are found on the postsynaptic membrane, and these are the metabotropic receptors. And the binding of a ligand, the neurotransmitters that we just talked about, to this postsynaptic metabotropic receptor usually causes the activation of an enzyme, and usually these are G-coupled, G-protein coupled receptors. Now, again, I'll show you that in a few minutes. As a result of its binding, this enzyme will become activated and will either cause an increase in the production or the loss or destruction of a second messenger system. And these different second messengers that may be activated or lost following the binding to these metabotropic receptors can include different second messengers like cyclic AMP. We could either increase or decrease cyclic AMP levels. We could increase or decrease cyclic GMP levels, or inositol 3-phosphate. All of these would be second messengers. And why they're second messengers, they act as an intermediary between the G protein and another enzyme system. So the second messenger, in turn, may activate other enzymes. It might activate kinases that phosphorylate, which we call phosphokinases, and these might be important to phosphorylate proteins in the cytoplasm or on the cell surface, on the membrane. And if you phosphorylate membrane proteins, often ion channels, this means that you might change the way that that channel works. And because these channels normally allow for the influx or the movement of ions, it may change the flow of these ions through those particular channels. Now, I want you to actually circle this particular slide because it's going to be important when we talk about sensory systems in the next lecture. So in lecture 5, you're going to see some of these in a little bit more detail. So hold on, we'll get to that a little bit later on, but just keep in mind, circle this slide off, make sure that you're clear on it. So these metabotropic effects are a little bit different. So ionotropic, it's almost immediate. So it's much more rapid. So it opens an ion channel after binding, and then you see the results in the cell almost immediately. The metabotropic receptor, when it gets bound or when a neurotransmitter binds to it, the effect on the cell, it may take a little bit longer. It may take time for it to become evident. And it's not necessarily going to change the membrane potential directly. It may all be internal. It may be activating these second messengers and different phosphokinases. But if you influence one of these different ion channels through this metabotropic effect or this metabolic effect, you might phosphorylate a channel, you might dephosphorylate a channel. The change in the membrane potential will then develop slowly as more and more of the second messenger and the kinases are brought online. So you may end up seeing an excitatory postsynaptic potential that is slower in response, so it's not going to be immediate, or you might see a slower IPSP, but again, these metabotropic effects take a little bit of time because all of these different enzyme steps have to be met, and there's a pattern and timing to all of these. All right, so again, we talked about this before, the importance of these metabotropic effects and how they might work. And as we take a look at the prime example of this G-protein coupled receptor, this would be the beta-adrenergic receptor or the beta-adrenoreceptor. And I've showed you a diagram over here. So this would be the beta-adrenergic receptor. It is a G-protein coupled receptor, so you can actually see it in profile if we're looking at it from the side, and this would be the cell membrane. The phospholipid bilayer denoted by these two different lines, and we would see that we have a classic 7 transmembrane domains. So here's one domain, number 2, number 3, 4, 5, 6, and then 7. So 7 different segments through the membrane. And then it's usually coupled with the G protein. In this case, it is a G protein called GS. And then this in turn, once this gets activated, after it binds to its ligand, here being noradrenaline, the G protein will then activate adenylate cyclase or adenylyl cyclase. Its activity is increased, and so we start to increase the production of cyclic AMP, and the cyclic AMP in turn will activate a kinase. In this case, it's protein kinase A, and it goes from an inactive form. In the presence of cyclic AMP, it becomes much more active, and this activated protein kinase A may then go on to phosphorylate other proteins, whether internally on the cytoplasm or maybe an ion channel. And this sort of sums up all the things that we were looking at earlier. And in particular, for the beta-adrenergic receptor, in this particular case with noradrenaline, the cyclic AMP, after going through all of these different steps, will then cause activation of protein kinase A, and this may in turn activate and phosphorylate the calcium channel. And as it phosphorylates it, it keeps it open longer, and so it changes the ion flow or the current through the calcium channel. And this is really important in terms of the heart muscle and its ability to change its contractility state. And you'll learn about that later on in the course. I think that's pretty cool. I hope that you understand all of these different steps. I'm hoping I'm not going too fast for you. And I'm hoping also that this concept of ionotropic versus metabotropic has been clear so far. All right, if you're still with me, let's just go on to the next slide. So just as we looked at for ionotropic receptors, we have a number of different ligands for metabotropic receptors, the same ones if you go back and compare. So here's the really confusing part, but I'm hoping it's not going to be confusing for you in this class this year. So acetylcholine, remember for ionotropic receptors, it was the nicotinic acetylcholine receptor. Here, this G protein, metabotropic receptor, is called a muscarinic receptor, and that's pharmacologically, it will bind to a substance known as muscarine, and it only activates these G protein coupled receptors that normally bind acetylcholine. And I use the term G protein coupled receptor. It's interchangeable with metabotropic receptor. These metabotropic receptors have a wide variety of ligands. So things like peptides can also bind, things like substance P, which is a neurotransmitter, beta endorphins, which hopefully we'll have time to talk about when we get into some cool things within the nervous system, antidiuretic hormone or ADH. We also have catecholamines, which will activate metabotropic receptors, things like noradrenaline, so the beta-adrenergic receptor that I just showed you. Dopamine is a classic example of a metabotropic receptor, and it is also characterized as a catecholamine. You might have heard of the neurotransmitter serotonin. Serotonin also acts on these metabotropic type receptors as well. And different types of purines, so DNA analogs like adenosine as well as ATP will also bind to metabotropic receptors. We also find them, by the way, in the nervous system, which is really cool. We have metabotropic receptors for ATP as well as adenosine within the CNS, which I think is like a really cool finding. Different gases like NO as well as carbon monoxide can also bind to metabotropic receptors as well. So again, really diverse, really cool, allowing for a lot of flexibility within the nervous system. And again, just to highlight the fact that these metabotropic receptors are really important. And so this might seem like a really weird sort of out of the blue slide, but here's why this slide is important. Because up till now, we have been looking at these different types of receptors, the two classes, ionotropic or metabotropic, and we've seen that they can cause differences in the postsynaptic potential. And now we need to see how all of this fits in together. So what we've been looking at so far is in the previous lectures 1 to 3, we've looked at how action potentials get generated and how they cause the release of neurotransmitters. And that was in lecture number 2 and number 3 in particular. And what we've looked at today, earlier today, is that on this postsynaptic membrane, we have these two different classes of these receptors, whether ionotropic or metabotropic. So why is that all important? Because for this next cell, this postsynaptic cell to respond, we need to know if it's going to be excited enough or not excited enough to make use of the information that is coming from this other neuron. And so if we were to put in an electrode into this cell near where we have the release of a neurotransmitter, so right here, so you see the electrode coming in, minus 70 millivolts at rest, the neurotransmitter gets released, it then binds to a receptor, a postsynaptic receptor. It's excitatory because it's depolarizing, so it's going up, and we know it's going to pass threshold. Well, again, this is a non-excitable membrane. They don't actually have a high density of voltage-gated sodium channels, so we can't generate an action potential. So this value of threshold here actually doesn't really help us very much. There are no voltage-gated sodium channels around to make use of it. So we have this really interesting signal being generated here, but if we see how this is spreading, because it's like getting the effect and it's not just staying in one area, this current, these ions are flowing on the plasma membrane of this cell. And if we looked at a further distance, we would see that what we started with has actually gone down in size. And we say the amplitude, the height that we're looking at here has gotten lower. And if we keep going, we'll see that this is actually getting smaller and smaller. Now, is that a problem? It may or may not be, because if it's still above threshold, by the time it reaches this trigger zone or this active zone, then this would be enough to trigger an action potential. And then this cell would indeed respond. And that's why we're so interested in these postsynaptic potentials. And again, taking a look at how this signal degrades and how it gets smaller and weaker, and if it will be at threshold by the time we get all the way here to the trigger zone, and if we can generate an action potential, is of great interest to scientists, but also to us as this course is looking at how this signal spreads from one neuron up here at the top to the next neuron that we're looking at right now. So I'm hoping that's a clear concept for you, why we're interested in these ionotropic, metabotropic receptors, because they're the ones that are going to influence the spread and the generation of these postsynaptic potentials. And again, we really want to make sure that this signal gets relayed from that cell to the next cell, and that's the whole purpose of these postsynaptic potentials. And if we're lucky and we get to above threshold at this trigger zone area where we have a high concentration of voltage-gated sodium channels, we will be able to generate an action potential. And that's why we've spent so much time talking about action potentials and voltage-gated sodium channels, and why we're so interested in the spread of a current on this plasma membrane. Again, hoping that's clear for you. I'm hoping these slides have helped to illustrate why we're so interested in these postsynaptic potentials. So the spread of these PSPs are of vital importance. It all begins after the binding of a neurotransmitter, and hopefully will generate some kind of postsynaptic potential. And then they're going to spread. This postsynaptic potential is going to spread from the source, from where it has bound the receptor, through passive conduction, because there are no voltage-gated channels. It's not going to generate action potentials. So in a lot of ways, this is kind of looking at the cable properties and the slow decay of a signal across this membrane, and hopefully it can get to the initial segment, very similar to the idea of a trigger zone. The initial segment or the trigger zone will have those voltage-gated sodium channels that we know are critically important and help to define what an excitable membrane is. Now, I'm hoping by this time, much of what we're talking about, you are developing a fluency in terms of neuroscience. I'm hoping you're talking like a neuroscientist. So I'm hoping that while you're sitting there at home, and you have no one else to talk to, but maybe your parents, that you're kind of talking to them a little bit about neuroscience and about the spread of action potentials. So today you get to really impress them by talking about these postsynaptic potentials as well. So here's this other thing. This is such an important biological phenomenon for one neuron over here to communicate with the next neuron and to get that signal to go across. with high fidelity, we want that communication to exist. And so as a result, what we often need to do is to help this postsynaptic potential. And these postsynaptic potentials are kind of unique. They can actually add together, which is why we're looking at this slide for postsynaptic potential summation. And because biological tissues, from lecture number 3, whenever you listen to it, whether it was an hour ago or it was a day ago or 2 days ago, these biological tissues have very, very poor conductive properties. They have very poor cable properties. So in your head, you know that meme where you have that lady kind of looking off and all of these mathematical formula going around? In your head, do you have those mathematical formula going on? My hands are disappearing online. Do you have all of those different formulas going on in your head? Is one of those formulas for cable properties related to the space constant or that length constant lambda? And do you see that little square root sign with the Rm over the Ri, the axial resistance? So we know that biological tissues have these poor cable properties. And this slide is designed to get you to think back to that particular slide. So we know that this current that we start off with gets smaller and smaller and smaller the longer this is going from where it has been initiated, and we want it to get to the trigger zone. And it's such an important phenomenon, and we need that signal to get to the trigger zone, that often it needs a little bit of help. So there are 2 different types of summation. One is spatial summation, and here we might have multiple inputs to the same neuron, and we might have 10 to 30 different synchronous IPSPs occurring all across the membrane, and each generated at a different synapse, and that will be enough to raise the signal above threshold to make it high above threshold at the trigger zone. And again, I'll show you that in a few minutes. The other way that we can do this is through temporal summation. So we may not have as many synapses, but the few synapses or the one synapse that we have can fire in succession really, really quickly, and it will generate these IPSPs at high enough frequency so that the summated potential coming from a few synapses might be able to reach threshold over that same small period of time. So it's a little easier to understand when we jump into some of these different diagrams. So here, for example, we might have 3 different synapses onto this cell. Okay, so hopefully that idea is one that you can now sort of visualize in your head. And actually, we don't need to visualize. It's right here in front of us on this particular slide. So here's this cell. It's receiving 3 different inputs, and we can see that each of these inputs is going to cause a small IPSP. So we'll get this from one synapse, we'll get another one from a different synapse, and this third synapse that we have here will generate this other IPSP. They're all coming in more or less at the same time, so synchronously, and that was an important characteristic. And if they are in synchrony, they're all kind of coming in at the same time, then we will be able to sum these net depolarizations into this larger depolarization. The cell sees the depolarization all at the same time, and so it looks like it's getting this massive depolarization. This larger synchronous, spatially summated depolarization, or IPSP, is much more likely to still be above threshold by the time it reaches the trigger zone than any one of these individual ones. Does that make sense? I'm hoping that it does. This concept is an important one for you, so I'm hoping that you'll spend the time to make sure that you know the difference between this example, spatial summation, and the next example, temporal summation. Another way to think about it, excuse me, is if you are running a fundraiser and everyone donates 50 cents, but you get a lot of people donating 50 cents at the same time, you may be able to reach your goal of getting $100 versus just like hoping that you'll get one large donation of $100. If everyone chips in like 50 cents all at the same time, then this is sort of an analogy for what spatial summation looks like. Temporal summation is a little bit different. Temporal summation is where we know these IPSPs on average last about 30 to 40 milliseconds before they start to decay. And then if during the time that we have generated an IPSP, we then get a second round of that same neuron firing, releasing more neurotransmitter before it started to decay, the next EPSP that's generated from that same synapse might actually allow for threshold to be reached at the level of the trigger zone. So it's adding on to a pre-existing EPSP. So whether it's like one neuron firing multiple times or a couple of neurons firing, a very small number of these synapses being active in a relatively short period of time will allow for temporal summation. All right, so hopefully those two ideas and these two different concepts are clear for you. Now, what we've been looking at is very unique in terms of talking about excitatory postsynaptic potentials, because generally we want action potentials to exist within the nervous system. But we also have this other interesting phenomenon of inhibitory postsynaptic potentials. And you might think that's bizarre. If you want action potentials, why bother having an inhibitory postsynaptic potential? At least I think it would be bizarre as a concept, but let's work our way through this. So let's first look at what an inhibitory postsynaptic potential actually is. So here's a really interesting phenomenon, and I also studied this as well. So when I had come back from Stanford and I wanted to do a little bit more clinical work and I wanted to work with an anesthesiologist, she was actually studying inhibitory synapses, and up until that time I'd been only studying the excitatory synapses. So it was a really unique opportunity, and I got to study these inhibitory postsynaptic potentials. And these inhibitory postsynaptic potentials are preferentially located on the cell soma, but in a unique position. So often we see these inhibitory synapses interposed about halfway between where we normally expect to see excitatory potentials being generated and that trigger zone, that active zone that we're looking at on those earlier slides. And because it's halfway in between, it could actually alter what happens at the level of that trigger zone. It can shunt or stop a depolarizing EPSP from traveling further down the cell. So it's blocking it, in other words, that EPSP spread. Now let me show you an example of what I mean by this. So here is an inhibitory synapse that's coming in. It will be releasing neurotransmitter, and here's an excitatory synapse releasing its transmitter. Again, it's not the transmitter that release that's important, it's the type of receptor that it's acting on, but here we're seeing the spread of an excitatory potential. Hopefully, it's going to be at above the level of threshold by the time it reaches this initial segment, but we also have this other inhibitory synapse that is here. And if it happens to fire pretty close to the time that this is spreading down, what might happen? So what will happen with this IPSP in terms of blocking this particular signal? And you can see that it is indeed blocking the progression of this signal. So this IPSP is a unique-ish situation. So if we go back to the very beginning of the lecture, so roughly about 25 minutes ago, we had talked about different types of channels opening. And oftentimes we see that for IPSPs, we involve a chloride channel. So IPSPs often involve the opening of a chloride channel. Chloride is usually higher outside the cell than it is inside the cell. It is a negative charge. And if we were to use that Nernst equation, we'd actually see that the equilibrium potential for chloride is actually very close to the value for the resting membrane potential, around -70. And we know that there's no activity normally associated with the resting membrane potential. So if opening of this chloride channel were to occur, there would be very little change in the membrane potential as a whole. And that's not what the situation that we're looking at. In that previous slide, what we're looking at is the spread of this depolarizing current. It's getting closer and closer to that trigger zone that we are interested in. But if we were to suddenly, halfway between where it started and before it reached the actual trigger zone, if we opened up a chloride channel, which would then cause hyperpolarization, it would block the membrane potential by bringing it back to -70. And that would mean that the depolarization would rapidly go back below threshold. And so we say that it clamps the membrane potential because it's very close to the resting membrane potential. It's bringing the membrane potential back down to the level that we would expect at rest. We're clamping it at that level. We're holding it at that level. In neuroscientists, we like to have our own lingo, and clamping just means that we're holding it at that membrane potential level. So we're preventing that depolarization. This is an inhibitory effect, and so this is a block of that progression. And these IPSPs are very strategically located on the cell body, on that soma, for a reason. We don't see them everywhere, and these inhibitory potentials are there to simply block these EPSPs, to prevent excitation. And again, you might be thinking, why? Why bother? And there's a couple of reasons, but let me go back and talk to you about one particular reason and talk about a general situation. So IPSPs, and therefore inhibitory postsynaptic potentials and inhibitory synapses in the nervous system are actually very, very important. And here's why. The general case that I mentioned to you a few seconds ago, within the nervous system, about 80% of all the synapses are indeed excitatory because our brain is geared toward activity. So generally when you're sitting here trying to make sense of what I'm telling you, or whether or not I'm going to go back and eat that strawberry because I'm really tempted to do that, your brain is being very, very active, and we get lots of EPSPs, we get lots of action potentials generated, and that's generally a really, really good thing. But if it's overactive, and this slide is actually from Epilepsy Canada, then we actually generate too much activity. Without that inhibition and without properly slowing it down, even though only 20% of the synapses are inhibitory, we actually have a condition that looks a lot like epilepsy. And that's overactivity of the brain. And you can imagine what that would look like in terms of being able to think properly or having all of your synapses being active at the same time. And that's not necessarily a good thing. And I know we like to talk about activity and excitation, but the inhibition is actually really important, which is why this bullet point is on this slide. In one scenario where we know that that's the case, where inhibition is generally lost in many forms of epilepsy or seizure activity, is again showing too much excitation and not enough inhibition. In future lectures, in other courses, you might hear about this excitation-inhibition balance, and this will hopefully remind you of that if you get to hear that again in future lectures. All right, so again, talking a little bit of now in this particular scenario, because we've been looking at postsynaptic potentials, raising things above threshold, and finding ways to block that potential from reaching that threshold, we've been looking at how these different forms of summation help in that process. But then why do we need to spend so much time thinking a little bit about this? This was kind of an Easter egg that I had buried earlier on, but we talked about action potentials and the frequency of action potentials giving your brain the ability to decipher different types of information. So what happens if you get a really, really strong, powerful synaptic input into this one postsynaptic neuron that's hoping to make sense out of all of this information that is bombarding it in the form of synapses and EPSPs that are being generated? What happens if you get enough of a PSP, an excitatory postsynaptic potential, that is not just above threshold, but is above threshold for a very long time, of over like 500 milliseconds? Can we make use of any of this to actually develop what we call a spike train? So depolarizing the trigger zone to threshold and then sustaining it, what does that mean for the nervous system, and how do we interpret this type of information? So if we keep it above threshold and we keep it depolarized, what would normally be expected to happen is that you would get one action potential, and then the voltage-gated sodium channels in that trigger zone area would be inactivated. And that's no good, because the absolute refractory period kicks in to play, and then the relative refractory period where some of them start switching back, and you won't be able to get another action potential until the membrane repolarizes. So after each action potential, which we call a spike, we would have one spike, then nothing else. We need to get that membrane repolarized or hyperpolarized as quickly as possible to restore those voltage-gated sodium channels to get ready because we're still above threshold. Can we still use that pre-existing signal? So while we have this large postsynaptic potential above threshold, we have to involve hyperpolarization to help generate this other action potential to recognize that we have a potential that is long-lasting and above threshold. And this means that we would be able to generate a spike train, but only if we have hyperpolarization. I'm hoping that's clear, and I'm hoping that the next slide will help to unravel that for you a little bit more. Just bear with me for a few more minutes. We're almost at the end. I'm kind of looking and eyeing that strawberry out of the corner of my eye. I'm hoping to kind of wrap things up, and hopefully that you're taking care of yourself while listening to this as well. And maybe we'll have a strawberry party at some time down the line. So this idea of a depolarization block, because this is what is going to happen. So let's imagine for a second that we have this threshold value, minus 50 or minus 55 millivolts, and we have these summated PSPs that are now reaching the trigger zone. It's above threshold, so we know that at the trigger zone, we'll generate this massive action potential, and then that's it. But we still have this other signal that's left over, and we're not making use of it at all. And this would sort of be the inactivation that we would normally expect, and we need to get it back below this threshold level, and yet on top of that, on top of this action potential that just got generated, actually the membrane is never going to get all the way back down because we still have that postsynaptic potential above threshold. And so this one action potential, it wants to reset all of the voltage-gated sodium channels in that patch of membrane, but it can't. That postsynaptic potential is now kind of working against it. So the one way that we can do this during this long postsynaptic potential, in order for us to be able to generate a second or third or fourth or fifth and generate what is known as a spike train, a whole chain of action potentials, and a spike is just an action potential, a range of these action potentials is to get rapid after hyperpolarization. We need to repolarize the membrane below the threshold value, so below the PSP level that is already here. Again, you need time to think a little bit about that, to process it. We can't get below this because the postsynaptic potential is continually depolarizing the membrane because it wants to generate another set of action potentials, but it can't because we can't get resetting. So it's a lot like a catch-22. And we can only do this by having voltage-gated potassium channels. And this is why they're so important at this trigger zone, because these voltage-gated potassium channels are the ones that actually help cause this after hyperpolarization. So they don't only repolarize, they will actually hyperpolarize, bring the membrane potential back down faster, and then also below the resting membrane potential. So this after hyperpolarization caused by these voltage-gated potassium channels, after each of these action potentials, while the membrane is still sitting there depolarized in this state, after a large postsynaptic potential, ensures that these voltage-gated sodium channels will be reconfigured, reactivated, and therefore the membrane excitability is going to be restored. So after hyperpolarization fades away, these potassium channels eventually close when the membrane gets repolarized. That membrane potential is now ready. It's still above threshold, and so it can actually now continually generate another action potential, which is why we saw that spike train. And so this is the only reason why we can do this. These voltage-gated potassium channels are incredibly important in this process, and I'm hoping that you understand why we talk about postsynaptic potentials, the different mechanisms that were involved, and we're at this review and summary slide. And here's a good sign for you to kind of go back, make sure that you understand all of these key points that are outlined here. And another really interesting fact is that if you look at this review and summary slide, I often will generate questions from this review and summary slide as well. So use these sort of as a guide for you to go back and study, not immediately after lecture, but maybe when you have time on the weekend. Also, again, just jotting down notes here is a wonderful idea. So I'm going to stop sharing my screen now. I'm going to go back here to this mode. So cheers. I'm going to have my strawberry now. Maybe I'll wait until a little bit later after. I feel weird and a little bit rude, and boy, my arms keep disappearing. Feel weird eating this now. But I do hope that you've been taking good care of yourselves. We're almost at the end of week 1. I know there's been a lot of information, and I'm hoping that you've been spending a little bit of time every day. Maybe it's 1 or 2 lectures at a time, and that you're not looking at it as something to be afraid of, but something that you can use to get motivated by and to teach other people about. I know that when I was an undergrad, I had a really hard time with neurophysiology. It was not necessarily my favorite topic in physiology. And I know that at times it can seem really boring and dry because we have to work up our vocabulary. But think back. Imagine like being able to understand what causes epilepsy and something really small, like understanding how shunting of a postsynaptic potential occurring and understanding how that might play a role in epilepsy. I think that's the coolest thing ever. And I hope that you do too. So I think I'm just going to end things here for now. Thank you again for being here, and thank you for really loving neuroscience, and I'm hoping that it's continually growing for you. I wish you good synapses. I wish you like time of rest and a time to eat strawberries. I'll see you in the next class, and hopefully this green screen effect will be a little bit better by the time we do meet for our next class. All right, so take care, everyone. Rest up those synapses. See you in lecture number 5.